Over recent decades, our knowledge of dietary fibre and available carbohydrates has expanded. This has led to a new physiological-based definition of dietary fibre, and subsequently new methodology has been developed, leading to AOAC method 2017.16 and ICC method 185 for total dietary fibre. In parallel, accurate methodology for the measurement of digestible starch, sucrose, lactose, isomaltose, glucose, fructose, and galactose has been developed. The method described in the Megazyme Available Carbohydrates booklet, and as detailed in this video, allows accurate measurement of available carbohydrates in all plant and food products, both solid and liquid, which is applicable in a variety of industries such as cereals, food and beverage, and animal feeds. This video tutorial will demonstrate the use of the kit with 2 cereal samples and the kit control. The kit contains sufficient reagents for 100 assays and is supplied with the detailed data booklet. The principle of the available carbohydrates assay is shown in this figure. Digestible starch in the food sample is hydrolyzed to D-glucose plus traces of maltose, and isomalto oligosaccharides are hydrolyzed to D-glucose and isomaltose using a mixture of pancreatic alpha amylase and amyloglucosidase. Maltose is hydrolyzed to D-glucose by maltase, and isomaltose is hydrolyzed to D-glucose by oligo-alpha 1,6 glucosidase. Sucrose is hydrolyzed to D-glucose and D-fructose by sucrase. And finally, lactose is hydrolyzed to D-glucose plus D-galactose by beta-galactosidase. In the presence of the enzymes galactose dehydrogenase and galactose mutierotase, galactose is oxidized by NADP+ to galactonate with the formation of NADPH. The amount of NADPH formed in this reaction is stoichiometric with the amount of D-galactose. It is the NADPH which is measured by the increase in absorbance at 340 nanometers. D-glucose and D-fructose are phosphorylated by the enzyme hexokinase and ATP to glucose 6-phosphate and fructose 6-phosphate with the simultaneous formation of ADP. In the presence of the enzyme glucose 6-phosphate dehydrogenase, glucose 6-phosphate is oxidized by NADP+ to gluconate 6-phosphate with the formation of NADPH. The amount of NADPH formed in this reaction is stoichiometric with the amount of D-glucose. It is the NADPH which is measured by the increase in absorbance at 340 nanometers. On completion of this reaction, fructose 6-phosphate is converted to glucose 6-phosphate by phosphoglucose isomerase. The glucose 6-phosphate reacts in turn with NADP+, forming gluconate 6-phosphate and NADPH, leading to a further rise in absorbance that is stoichiometric with the amount of D-fructose. Prior to sample analysis, prepare required additional reagents as described in the kit data booklet. Subsequently, the kit components should be prepared as described in the kit data booklet, and once prepared, they are ready for use in the assay procedure. Use the contents of bottles 1, 4, 5, 6, 7, 8 and 9 as supplied. The contents of bottle 2 are dissolved in 12 millilitres of distilled water. The bottle is capped and the contents are mixed thoroughly to ensure complete dissolution. Transfer this to a polypropylene tube and store below -10 degrees Celsius between use. Accurately weigh 0.5 grams of pancreatic alpha amylase and amyloglucosidase mixture and record the weight. Add the enzyme mixture to 25 millilitres of 50 millimolar sodium maleate buffer, pH 6, and dissolve by stirring on a magnetic stirrer. Alternatively, and preferably, add 2.5 grams of pancreatic alpha amylase and amyloglucosidase mixture to 35 millilitres of distilled water and dissolve by stirring on a magnetic stirrer. Weigh 17 grams of ammonium sulfate. Add this to the dissolved pancreatic alpha amylase and amyloglucosidase mixture and dissolve by stirring. Adjust the volume to 50 ml with 50% weight per volume ammonium sulfate solution and store in a cold room. This suspension is stable for up to 3 months at 4 degrees Celsius, or for several years if stored below -10 degrees Celsius in polypropylene containers. Accurately weigh approximately 0.5 grams of sample into a 40 ml polypropylene tube and record the weight. In this demonstration, we are using 2 serial samples in duplicate and the provided kit control in duplicate. Add a 20 by 6 mm stirrer bar to each tube. Wet the sample with 0.5 ml of 95% ethanol and add 17.5 ml of 50 mmol sodium maleate buffer pH 6 to each tube. Place the tubes in the polypropylene holder shown on a 2 mag mixed drive submersible magnetic stirrer in a water bath and allow the contents to equilibrate to 37 degrees Celsius over 5 minutes with stirring at 170 RPM. Add 2.5 ml of pancreatic alpha amylase and amyloglucosidase solution to the tubes and incubate the reaction solutions at 37 degrees Celsius with stirring at 170 RPM on the 2 mag mixed drive submersible magnetic stirrer. Alternatively, if using an ammonium sulfate suspension of the enzyme, add 1 ml of enzyme suspension and a further 1.5 ml of sodium maleate buffer. Cap the tubes and incubate for exactly 4 hours at 37 degrees Celsius. Near the end of the 4-hour incubation, add 25 ml of distilled water to label tubes. After exactly 4 hours, remove the incubation tubes from the water bath and store them at room temperature for 5 minutes to allow solids to settle. Remove the caps from the tubes. Accurately transfer 1 ml of the reaction solution to the label tubes containing 25 ml of distilled water. Alternatively, after 4 hours, filter the contents of each tube through glass fibre filter paper. Transfer 1 ml of the filtrate to 25 ml of distilled water in label tubes. Cap the tubes and mix the contents thoroughly using a vortex mixer. If not analysed immediately, store these tubes at 4 degrees Celsius. transfer 2 ml of these solutions to labelled microfuge tubes. And centrifuge at 13,000 RPM for 5 minutes. Follow the assay procedure as described in the data booklet. In this demonstration, we will use the prepared cereal samples, kit control, and the D-glucose, D-fructose and D-galactose standard solution, bottle 8, along with an associated blank test tube. Accurately transfer a 0.1 ml aliquot of the centrifuge controls and samples into their corresponding test tubes. Pipette 0.2 ml of 50 millimolar maleate buffer pH 6.5 containing 0.5 mg per milliliter of BSA into the control and sample tubes. Swirl to mix the contents of bottle 4, sucrase, maltase, beta-galactosidase and oligo-alpha-1,6 glucosidase prior to dispensing. Accurately pipette 20 microliters of suspension 4 into the bottom of the control and sample tubes. And incubate for 30 minutes at approximately 30 degrees Celsius using an external heat block. During the sample incubation period, prepare the kit standard tube by the addition of 0.1 ml of the contents of bottle 8, D-glucose, D-fructose plus D-galactose standard solution into the standard test tube. After the 30-minute sample incubation, accurately pipette 2 ml of distilled water to all assay tubes. Pipette an additional 0.22 ml of distilled water into the standard assay tube. Pipette an additional 0.32 ml of distilled water into the blank assay tube. Pipette 0.1 ml of the contents of bottle 1, imidazole buffer, into all assay tubes. Pipette 0.1 ml of the contents of bottle 2, NADP plus and ATP, into all assay tubes. When all of the components have been added, mix the tube contents thoroughly and incubate the tubes at 30 degrees Celsius for approximately 3 minutes. After 3 minutes, record the first absorbance reading, A1, at 340 nanometers for all of the assay tubes. In this demonstration, we are using the Megaquant Wave spectrophotometer, set to read at 340 nanometers, in conjunction with an external heat block to achieve the correct incubation temperature. Alternatively, a temperature-controlled recording spectrophotometer with 1 cm path-length cuvettes can be used. Swirl to mix the contents of bottle 5, galactose dehydrogenase and galactose mutterotase prior to dispensing. After recording the A1 absorbance value, pipette 20 microliters of the contents of bottle 5 into all assay tubes. Mix the tube contents thoroughly and incubate the tubes at 30 degrees Celsius for approximately 3 minutes. After 3 minutes, record the second absorbance reading, A2, at 340 nanometers for all of the assay tubes. Swirl to mix the contents of bottle 6, hexokinase and glucose 6-phosphate dehydrogenase prior to dispensing. After recording the A2 absorbance value, pipette 20 microliters of the contents of bottle 6 into all assay tubes. Mix the tube contents thoroughly and incubate the tubes at 30 degrees Celsius for approximately 5 minutes. After 5 minutes, record the third absorbance reading, A3, at 340 nanometers for all of the assay tubes. Swirl to mix the contents of bottle 7 phosphoglucose isomerase prior to dispensing. After recording the A3 absorbance value, pipette 20 microliters of the contents of bottle 7 into all assay tubes. Mix the tube contents thoroughly and incubate the tubes at 30 degrees Celsius for approximately 10 minutes. After 10 minutes, record the fourth absorbance reading, A4, at 340 nanometers for all of the assay tubes. This absorbance reading completes the available carbohydrate assay procedure. The relevant absorbance readings A1, A2, A3 and A4 of the sample and the blank reactions are used to calculate the D-galactose, D-glucose and D-fructose concentrations in the original samples. When performing this test using the pre-installed protocol on the MegaQuant Wave spectrophotometer, the results will be automatically calculated and printed via the onboard printer, or the data can be exported to a computer using the SF Capture software. Please see our MegaQuant Wave video for further details. If the results output are raw absorbance values for both blank and samples, the calculations of available carbohydrate content can be performed manually as described in the calculation section of the kit booklet. Megazyme has also developed specific Excel-based MegaCalc applications for each Megazyme kit to allow quick and easy results analysis. Results can be analysed using the MegaCalc application specific to this assay kit, which is available to download free of charge from the Megazyme website. Results can be calculated on an as-is basis, or moisture content of the sample can be allowed for. The MegaCalc spreadsheet provides full instructions for use. Open the MegaCalc worksheet and input the following. Sample details. Absorbance readings for the blanks. For each sample, input the sample identifier. If a liquid sample has been used, input the sample volume used. For solid samples, alter the sample weight, if a weight other than the default 0.5 grams has been used. Alter the extract volume if a volume other than the default 20.5 milliliters has been used. For each sample, input the absorbance values recorded. Alter the sample volume if a volume other than the default 0.1 milliliters is used. When all of the data has been entered, the concentration of D-galactose, D-glucose and D-fructose in the sample is automatically calculated and given as grams per litre in the original sample for liquids, or as grams per 100 gram on an as-is basis for solid samples. If the moisture content of the samples is known, input the moisture content percentage. When this additional data is entered, the concentration of each analyte is automatically calculated and given as grams per 100 gram on a dry weight basis for solid samples. Available carbohydrates are calculated as the sum of D-glucose plus D-fructose plus D-galactose. For further information, the relevance and measurement of resistant starch, digestible starch and available carbohydrates are discussed in detail in a paper by McCleary et al.